Hello, everybody. I would like to warmly welcome you on today's webinar, where we will focus to utilization of artificial intelligence in the correlative microscopy, especially or particularly AFM-in-SEM. My name is Jan Neumann. I'm CEO and co-founder of the company Neno Vision, and it will be my great pleasure to talk and focus on the correlative microscopy and AFM-in-SEM part. I would like to welcome here also Jiří from Machine Learning College.
Thank you for having me here. Hello, everyone. My name is Jiří Materna, and I will focus my talk basically on the principles of machine learning and also on some applications of deep learning in microscopy.
The scope of today's webinar will be following. On the beginning, I will briefly introduce you Neno Vision, also AFM in SEM and generally correlative microscopy approach. Then I will come to the stage Jiří, and he will give an introduction to deep learning in microscopy approaches. Then I will take over again, and I would like to share with you some application examples of utilization of artificial intelligence, especially in AFM and SEM correlative microscopy. And by the end, we will have sessions where we will answer some of your questions and we will comment it. During the whole webinar, please feel free to raise any questions in the dedicated tab. My colleagues are ready to reply already during the webinar. As I mentioned, as a first part, I would like to briefly introduce our company and also the technology combining AFM and the SEM. Our company Neno Vision was founded in 2015, and we are located in, we say, heart of the Europe, in the Czech Republic, in the city Brno. We are quite proud of this location because the Brno is a university city, and it's also historical world center of electron microscopy. Companies like Thermo Fisher, Toscano, DeLong Instruments develop and produce their electron microscopes here in Brno, and therefore we are part of this microscopy community. Mission of the Neno Vision since the very beginning is very straightforward. We are established to create and develop the unique tools on the top technological level, which helping to drive scientific discovery with a significant impact. And therefore, the innovations, we're also using the artificial intelligence is natively inherent in our DNA. We came up in 2016 with our first product, LiteScope, which is a very small, compact, and high-performance AFM, which you mount inside your electron microscope, and therefore we merge the forces of both, of AFM and the SEM. We believe in cooperation with the community, because we understand that together with other partners, we are able to achieve much more important and significant results, and therefore I would just highlight a few of our partners, with them we have developed the cooperation in the last years, like BASF, Thermo Fisher, Toscano, or ETH Zurich. With them, together we are focusing to current trends in the microscopy as we see it, such as in-situ microscopy or multimodal and correlative sample analysis, but definitely also utilization of artificial algorithms in microscopy. And why we want to do that? Because we feel our obligations to help to solve some of the global challenges. So together with partners, we are helping the solutions which would help to improve the energy storage, like batteries or production of the semiconductor devices, or the development of novel, suitable, sustainable materials. Together with them, we believe that we are able to break the limits of in-situ multimodal correlative characterization. Now I would like to briefly describe and introduce you more about the AFM and the SEM technology and introduce our product LiteScope. So why we want to combine AFM with the SEM together? Its answer is pretty simple, because AFM and SEM are complementary. With the AFM, you are able to get very high resolution 3D topography. You can analyze mechanical, electrical, or magnetic properties. And on the other hand, we can utilize the advantages of SEM. It's like very fast, large scale, relatively large scale 2D imaging. You can do the chemical analysis, you can do the surface modification, and combination by these two techniques we see as a very powerful and promising field. We see three main benefits why to combine these techniques together. The first of them is in-situ analysis. The second, we are able to achieve the complex and correlative analysis at the nanoscale. And the third is a very precise and fast localization of the region of interest. With that, we are providing the scientists the information about their sample, which otherwise are not unable to acquire, or it's extremely complicated. So what do we understand if we say in-situ correlative microscopy? It means that together with both AFM and the SEM, you are able to measure and perform the analysis at the same time and the same place and under the same conditions. And therefore, there is no need for a sample transfer between the different instruments. And what I really love when our scientific ambassador, Mark Willinger from ETH Zurich, say the LiteScope is like a Swiss Army knife inside your SEM. So this is how our instrument is exploring the possibilities and capabilities of in-situ correlative analysis. The second area, it's a complex and sample analysis. And here we talk about the multi-dimensional or multimodal correlative imaging using AFM and with the SEM. As an example, I can show here on these 2D materials where we can acquire the high resolution topography. We can add directly the information exactly from the same area about the local hardness. We can add the information from the electron microscopy, and also we can add the chemical composition coming from, for example, from EDX. And this is complex and correlative sample analysis as we see it. And the third main benefit, which we see as a fast and extremely precise navigation of AFM tip to the region of interest. And you can see it on the video. It's pretty simple. You can use a SEM for the navigation of AFM tip to the specific area. So in this case, you see the AFM tip, how it's approaching on the protein, on specific protein, a specific position where we can perform additional characterizations. As I mentioned, and I think it's clear, so our focus is for correlative microscopy, and I would like to introduce also our CPEM technology. CPEM stands for Correlative Probe and Electron Microscopy, where we combine both techniques together. So on this video, you can see the AFM tip. You can see the electron beam focus very close to the AFM tip. And then during the measurements, we start to scan with the sample. Therefore, electron beam, also the probes are static. And during the analysis, we are collecting different information from both instruments. There is a constant offset between both images, which can be easily removed. And by the end of the day, we are ending with the correlative CPEM view. If you go to our NenoView control software, in this video, you can see how easily you can simultaneously collect not only just two, but several informations from about your sample. So on this one video, inside our software, you see the AFM topography, you see the signal from secondary electron, from backscattered electron, also the channel related to the local elasticity, and all of these signals are acquired simultaneously. we are bringing the different modalities with the AFM into the SEM characterization possibilities. So with our AFM, you can characterize a variety of mechanical properties like topography, elastic properties, sample hardness, etc. You can analyze also several electrical properties when you can make a local conductive mapping, or you can measure surface potential or measure IV curves, etc. You can characterize the piezoelectric properties of your sample, or you can analyze the magnetic properties. So I guess you can imagine that if we have several signals and images together, that utilization of the machine learning algorithms helping with the understanding and interpretation of the data is crucial. Of course, all of that can be applied in different application areas. One of the most important we see in the material science, where we already perform many interesting measurements on 1D or 2D materials, on steel alloys, batteries, ceramics, etc. You can utilize it in analyzing of different nanostructure, for example, in situ modified surfaces by the focus ion beam or gas injection system, or to make the characterization of quantum dots or some nanopatterns. It has application in semiconductor industry. Here I would just highlight the failure analysis, for example, or we have shown the advantages also for life sciences like the cell biology. With that, I would like to conclude and just give you the introduction about the AFM-in-SEM technology and possibilities. And with that, I will pass my word to Jiří, and he will focus more on deep learning approach in microscopy.
All right. Hello again. My name is Jiří, and before we start with the talk itself, I would like to tell you a few words about Machine Learning College, which is a private company focusing on machine learning trainings and applications of machine learning in many fields. This is the company which I founded 6 years ago, and where I've been working as the teacher. And this company is offering mainly trainings and workshops in the AI fields. We offer the courses privately or publicly. It depends on the type of the course. And we also offer consulting and solutions in the field of machine learning and artificial intelligence. So if you are interested in a training, either online or on site, you can check out the information at our web, mlcollege.com. And now let's start with deep learning and the stuff I would like to present you today. So first of all, it would be great to distinguish between three terms: deep learning, machine learning, and artificial intelligence. Artificial intelligence is the most general term covering everything. There are many principles and many algorithms implementing artificial intelligence. For example, machine learning, which is the most important one today, but there are many others like expert systems. Machine learning is a very big field full of many algorithms and approaches, but one of the most probably the most well-known approach is an artificial neural network. There are many other algorithms like random forest or logistic regression or support vector machines, but today we are going to talk about artificial neural network. And if the neural network has some specific properties, like that it is big in some sense, meaning it has many layers, then the method is called deep learning. And this is the stuff we are going to focus today on. So we should start with the description of a neural network. From the mathematical point of view, a neural network is just a function which maps objects from n-dimensional vector space of real numbers to m-dimensional vector space of real numbers. It is pretty useful for our application because every image can be represented as a vector in n-dimensional space. So specifically, artificial neural network can be used for mapping images to images. But we can also map images to some other representations like vectors of classes or some numerical predictions, what we want. But the main thing that we can use an image as the input to the artificial neural network. Before we start talking about the network, we should talk about a single neuron. If the network consists of just one neuron, this neural network is called Perceptron, and it implements this simple function. It takes n numbers as the inputs, and it produces one number A. When we want to compute the output based on the input, we simply multiply the inputs with some weights. Here you can see weights W1, W2, W3, and Wn. So we multiply the inputs with these weights. The weights are just real numbers, and so we get n numbers. Then we sum them up and get one number Z. And we are almost done. The last thing we have to do is to transform this number using some activation function. Activation function is a simple function. It can be any differentiable function, for example, a linear function or sigmoid function or hyperbolic tangent. These functions are simple functions, and the main purpose of these functions is to make the model nonlinear. So this is one neuron. If we combine the neurons into a network, we can get a model like this. This is a network of neurons where we have 4 inputs, x1, x2, x3, and x4, and 3 outputs, y1, y2, and y3. You can see that the neurons form so-called layers. So this is the first layer, and you can see that, for example, this neuron takes all inputs from here, and it produces one output. It is here. Each neuron in the first layer has a single output, and this output goes as the input to the next layer. And this is the principle of this architecture of a neural network, which is called feed-forward network. I should go back to this model where you can see the weights. They are the most important part of the neuron because these are the only parameters which are tuned during the training. What does it mean, training? Well, we want the neuron to produce some useful information, some useful output. So we have to tune these numbers, these parameters, to produce some useful outputs based on the inputs. For example, I'm going to show you an example of classification where we want to classify some images into classes. So the output here can be the class, and we want this output to correspond with the actual output. So when we are training the whole network like this, we have to tune the parameters of the network. And because we want to get some desired output based on the inputs, we have to set the parameters, the weights, in a proper way. And this is done during the training. You can see the training as an optimization task where we have an error space in many dimensions. Here we have only two dimensions, two parameters, W1 and W2, but in practice it can be billions of parameters, and we want to find the minimum of this error function. And this minimum corresponds to a combination of the parameters of the weights that are best, or one of the best solutions for the problem we are solving. Here we can see the pipeline of the most important task in machine learning, which is supervised training. In this case, it is classification. At the input, we have examples of images of apples, different images of apples, and also different images, various images of cupcakes. And this data is called training data. They are used for training, and the very important thing is that for each example, for each image, we know what is the class. For these images, it is apple. For these images, it is cupcake. And we use this training data to train the model, to tune the parameters, the weights. Of course, we want to train the network such that for images of apples, the output will be an apple, and for images of cupcakes, we want the network to produce the output corresponding to cupcakes. So we train the model for many iterations, and once the training is done, we can use this network, this trained network, for predictions on unseen and unlabeled data. So we can feed the network with some unknown image, and the goal of the network is to predict the correct class, and we hope that the class is correct. So this is the pipeline of training and predictions, which is typical for neural networks. And now I'm going to show you some examples from microscopy where neural networks and machine learning can be applied. So the first example is denoising. On the left-hand side, you can see an image from scanning electron microscopy. You can see that this image, it is some nanostructure. This image is quite blurry and full of noise. So how to improve this image? Well, one of the reasons why the image is noisy is that high energy was used. So we can improve the result by application so-called scanning low energy microscope, electron microscopy, and the result would be like this. It is much better, but still it is not perfect. So now there is a place for application of deep learning. We can train a neural network which is trained to remove the noise and the blurriness. And if we train properly such a model, and if we apply it on this image, we get this result. So you can see that it's much better than the original one, and this task is called denoising. Another application is image segmentation. The task is to identify some regions on the image. So in this case, on the left-hand side, you can see an electron microscopy image of steel, and the task is to detect regions corresponding to some contamination. So we would like to get this output, and it can be solved easily using machine learning and artificial intelligence, such that for each pixel of the input image, we want to predict the class, and the class can be either steel or the contamination. And of course, there can be various number of types of contamination. So it can be not only a binary classifier, but we can classify the input image into many classes. So this is called image segmentation. The last example of application of deep learning in microscopy is superresolution. On the left-hand side, we have an image in low resolution, and the goal is to increase the resolution. In this case, we increased the resolution 4 times. Well, the task is pretty well defined, but it's not really easy, because if we want to create an image in high resolution, there is missing information. We have to create some pixels which were not presented in the original image. So it's not pretty easy. And we typically use state-of-the-art architecture, which is called generative adversarial networks. This architecture consists of 2 networks. The first one is generator. It is the bottom of this image. On the input, we would have the original image in a low resolution. In this case, we have, you can see some d-dimensional noise vector. It is for a different application, but for our task, it would be an image in low resolution. And the generator, the task of the generator would be create an image in high resolution. But as I told you, there is a lot of missing information, and it is not it's not an easy task. And it sometimes happens that the result doesn't respect the information which is hidden in the input image. So it can create something which is not presented in the original image. So it is reasonable to have some discriminator network, which is here, which controls the output here. It predicts. It is a classifier which tells us if the output is correct or not, and gives this feedback back to the generator network. So it controls the outputs, and using this architecture, we are able to create much better results. On the last slide, I will show you some results of this network, of this architecture on improving images of faces. On the left-hand side, here you can see the input and various number of different approaches to improving these images. One of the state-of-the-art models produces this result. But for example, here you can see the problem which arises when we use this GAN models. As I told you, it is necessary to create some new information, but it can happen that it adds something which is not presented in the original one. So, for example, in this case, you can see that from the image of a little girl, it created an image of a young lady. In this case, it is probably an image of an old woman. It modifies it to a man, and in the last example here, it removed mustache of this man. So we have to be really careful about this model, and the main purpose of the discriminator here is to control the modifications, and the goal is not to allow to create such modifications. So it is possible. It's not easy, but it's possible, and it produces really good results even on microscopy images where we require to preserve the information from the original image. And that's it. Thank you for your attention. And if you are interested in some trainings in machine learning, you can find all information on our web, mlcollege.com. Thank you.
Thank you very much, Izzy, for really nice, interesting, and inspirative talk. Let me continue to share with you some another application examples utilizing artificial intelligence in combination of AFM and SEM. And particularly, I would like to mention that it's using the artificial intelligence, we have to be always really careful about the results, as Jiří mentioned. And for us, we currently see it as a more guide, if you are driving the car, the artificial intelligence helping you to drive the car, but it's not independent. So I think it's very similar in the science. And so to show you some examples where we use artificial intelligence, I will come back to our CPEM technology where we are simultaneously acquiring the AFM and SEM data, and resulting with the two images coming from the SEM and AFM. The images therefore needs to be correlated and put together. We do it manually in the way that we take the topography from the high resolution topography from the AFM. We fit the SEM image exactly to the correct area of interest, and then we end up with a really nice and correlative CPEM view. In the last year, we have developed the CPEM Plus technology, which is the functionality utilizing the simultaneous acquisition by AFM and SEM data using CPEM and combine it with the AI algorithms. In this way, we're able to prepare the functionalities which will automatically correlate the images. It's a fast, it's a precise, and also it simplified the correlative data evaluation. This work was done with cooperation with the company Science Desk, and it's a part of our software control unit, NenoView software. The CPEM Plus, which is an AI-driven correlative functions, are doing basically two things. The first is they automatically remove or correlate the offset between AFM and the SEM images, as you can see it here on the right side. And in some cases, it does not fit perfectly, because in some measurements we see the drift of electron beam during the measurements. Therefore, we adjust also the artificial intelligence functions algorithms in this way, and we were able to optimize the algorithms in the way that it corrects such a electron beam drift. So in traditional workflow, we are setting up our CPEM view, we collect AFM and SEM images, and then we automatically adjust and correlate both images together, resulting with the ultimate precision correlation between both examples. All these functions are integrated inside our NenoView software. So here on this video, you can have a look how it's done. So in our browser, you can load the data where contains the AFM and SEM data, and then you would like to try to correlate them. This is on this image. You can see the difference and the shift of both images, and then you just apply the AI-driven functions, which will automatically correct the offset or also offset with the drift. Then you just select the different channels and layers which you would like to store, and to the file there are additional new layers already cropped together and aligned together are stored, so then you can work with them later on. All basic or fundamental channels are already present, so always you can continue. So in this way, we guide and simplify the workflow and guide the user during the data processing. I want to show you two examples. So this is a utilization of CPEM Plus on polished metallographic section. So again, we have a SEM and AFM images. Here you see when they are automatically corrected, but again, I'm selecting the images where there is a visible drift of the electron beam. And again, once we apply the yeah, we can have a look into more details. And once we again apply the algorithms, also utilizing the drift correction, you can see on both details that we were able to much better correlate both images together. I would like to show one more example, which is actually the FIP pattern magnetic domains, where you have a thin films, you use focus ion beam to change the magnetic domains. And what is very interesting, that we have a completely different SEM and AFM image. So for the human being, it's very complicated to correlate such an image. But with the well-trained artificial intelligence network, it can be done. And again, here you can see just the offset removal. But again, when we applied also the drift correct function, you see on the details that the network were able to very precisely match both images together. So also sometimes the neural network see much more than the human beings, because they are focusing on different aspects on the images. And the next example, I would like to show you the results of our current work with the Brno University of Technology and Faculty of Information Technology group of Professor Harold, when we wanted to automate the AFM images and reconstruction. We just recently submit the paper, so I'm sure that pretty soon you will be able to read whole paper by your own. So what was the aim? We aim to fully automate AFM image processing and also then the image reconstruction. As you probably well know, AFM imaging simply has artifacts, because we are scanning with the physical tips over the sample, and therefore within the imaging we see the tip convolution. There are plane or line leveling. There is a noise. Usually you can use a standard software tools like a video or a digital surf to work with such artifacts, but we are searching for the automated approach which would simplify this data processing. And also additionally, what is a very challenging, and it was also mentioned by Jiří, for the training of such AI algorithms, you usually need quite large training data sets. And it's very problematic for the AFM microscopy technique, but not only, because it's extremely time consuming. You have a variety of the samples. So how we approach that? So first of all, we came with the first trick, that on the beginning and as the inputs to the neural networks, we use two images for backs and forth scanning, which means we have two images visualizing the same object. Then we were able to create the synthetic training data, which were used for the training of the neural network. And this training data already inherit the artifacts which are typical for the AFM imaging. And using that, we generate the large data sets with the ground state, which means something what we think it's a reality, and then how the visualization should look like from the left to right scanning direction and right to left scanning direction. We train this, we use this data for the training of the network, and by the end we are coming up with the data reconstruction. Simply we use AFM data as an input, and we reconstruct the image as an output. So if you look on the performance, so we compare the manual image processing for the experienced user and automated image processing. So we have three examples: wafer, some cells, and silicon. I think it's microsilicon. Always in the bigger scale and then in the details, and we can compare what a human being would do with the baseline and averaging image processing, and then the outputs from the train network. And you see that the neural network were able to very well correlate with the work of the manual experienced user. So therefore, with automatic manner, we can eliminate traditional or typical AFM artifacts. Therefore, we see the huge benefits in this approach. Since it's a time efficient and automated workflow, it does not require the expert or experienced user for this image processing. You don't need any programming knowledge or AI knowledge, and still we keep all the original data available, so you can do always the cross-check. And with that, I would like to just say that Denovision and also our LiteScope is ready for the era of artificial intelligence, which is actually part already of the AFM, of our AFM in SEM solutions. All the computing which is required for such operations is already done in the NenoBox control unit, and AI is controlled via our software. We have developed we call it Nenovision AI framework, simply because we are able to add their additional algorithms, additional functionalities, which would contain the specially trained networks. And also we are able to open this to the community or for the customers, which can train and test also their algorithms. And for the third part, we are ready to have an adjustable computer power, because as you know, as you might know, different algorithms might have a different requirements for the GPU processors, and we have a kind of modules offering different computing power, which can be directly integrated into our control unit. So with that, I would like to conclude my part. If you are interested about more information or about our product, just feel free to contact us, contact our global partners or us directly. You can also go to our webpage where we have a lot of information. You can follow us on the LinkedIn, Facebook, etc., or on the YouTube account where we have a lot of webinars available, and you can gain more information or the specific application according to your need. If you want to meet us personally, soon we will be in M&M in Oregon in US, or MCM conference here in the Czech Republic, and we'll be very happy to meet all of you and discuss actually the needs and opportunities for the artificial intelligence in the microscopy. So with that, I would like to shortly summarize. I want to introduce you our LiteScope, which is compact and easy to use AFM in SEM. We see the huge benefits for the in-situ characterization, complex and correlative data analysis, and also fast and easy localization of region of interest. Jiří explained you the basics about deep learning, and he presents some examples and deep learning algorithms in microscopy. So he was showing how it can be used for the denoising, image segmentation, or super resolution. And by the end, I was trying to show you how AI technologies can be used in the AFM-in-SEM technology. I was presenting our CPEM Plus technology, or automated AFM image processing and reconstruction algorithms. With that, we are almost on the end. So we have several questions here, which we would like to maybe comment and answer. So hello again. So we will do it lively so you can see us. So I will start with the first. So the question is, how is it compatible? How is it compatible with all SEMs? So I guess the question is targeting to the AFM. And yes, with the majority of the SEM, you can place our AFM into all manufacturers or main brands. like Thermo Fisher, Tescan, Zeiss, Geol, or Hitachi. The main limitation, almost only the limitation, is the chamber size of the chamber. So if you want to cross-check the specific SEM, just let us know. We have a list of the suitable SEMs and we will reply directly. Maybe I will.
Okay, I have only one question for me, which has actually two parts. So the first part is how many training examples were necessary for your GAN to improve microscopy resolution? And the second part is how we created or where we got the pairs of low resolution and high resolution images. So the first question, the first part, how many training examples? We created only, let's say, 100 or maybe less very high resolution images. Then we cut the images using a grid, so we get smaller images, but still in a higher resolution. So using this technique, we created like hundreds or maybe thousands of images, and then we downsampled these images into low resolution. So it is pretty simple technique, which guarantees that we have exactly the same image in high resolution and low resolution, which is necessary for the GAN algorithm.
Yes, thank you. So I have an additional question. Can your device improve the resolution of SEM? If you have not so good SEM, is it better to buy a new SEM or just buy your device? So it's also, of course, depending on your application. What we do generally, AFM has an ultimate resolution. So yes, if you place our AFM inside your SEM, you will get a very high resolution AFM images. We will not improve that much the SEM. So, but I cannot just advise if you should buy the new SEM or not. What I can advise, please let us know. We can discuss exactly what's your interest, what you want to do, and then we can together end up with the, I would say, the most suitable solution. Can you move the tip and the SEM beam in the synchronization? It seems very difficult as the different SEMs will have a different drivers. So maybe there is a bit of misunderstanding when I was talking about our CPEM, where we are simultaneously acquiring AFM and SEM data. There is a trick. We are not synchronizing AFM and SEM scanning. Both during the measurements, they are static. They are not moving, and we are scanning and moving with the sample. So the sample is moving, and so there is no need for any synchronization between AFM and SEM. Maybe I should mention it or highlight it a bit of more, that in the SEM community, everybody is used to scan with the E-beam. In our special CPEM measurements modes, we are not scanning with the E-beam. E-beam is static. We are scanning with the sample. And therefore, it's pretty straightforward to make such measurements in any kind of the SEMs. So with that, it was a few questions which we highlighted. So with that, I would like to thank you very much for your staying, for watching us. We are happy that we have such a strong community that is interested about the microscopy and also utilization of the AI. Please be with us, communicate with us, and we'll be very happy to move this area or even farther with you. Thank you very much for your attention, and I wish you have a nice day.
Thank you.